Hey everybody, this is our first lesson. We're going to try with videos so that while I'm away with the sub you'll be able to still learn the information from me. So we're going to do this one on DNA and its structure. You can see at the top DNA stands for deoxyribonucleic acid. So the D is for the deoxy, the N is for the nucleic acid part. Now, all nucleic acids, you remember, are made of nucleotides, right? So nucleotides are what you see in this picture here. So nucleotides. All right, so nucleotides are all made of a phosphate, a sugar, and a base. All right, you should know from this point on, no matter what, forwards, backwards in your sleep, phosphate, sugar, base, phosphate, sugar, base, phosphate, sugar, base. From here on out, whenever anybody asks you, what are nucleotides made of, you say a phosphate, a sugar, and a base. Now, this is a generalized structure of a nucleotide. This pentose sugar, penta, of course, means 5, meaning that this is a 1, 2, 3, 4, 5 carbon sugar, okay? But the sugar here matters. Pentose is a general name for the sugar. In DNA, this sugar is deoxyribose. Okay, now notice deoxyribonucleic acid, right? DNA gets its name from the sugar, deoxyribose. That's a part of its nucleotides. So DNA, deoxyribonucleic acid, is because the pentose sugar in DNA is deoxyribose. The other part of the nucleotide is this base, this nitrogen-containing or nitrogenous base. There are 4 possible bases, okay? The first is adenine, which starts with an A. The second is thymine. The third is guanine, and the fourth is cytosine. Now notice I'm just writing down the letters for these bases, okay? That's because that's all you're ever going to have to know. Adenine, thymine, guanine, cytosine are usually referred to as A, T, G, and C. So that's a nucleotide, and these nucleotides are going to get strung together to make the DNA molecule. Before we go any further, you do have to know that there is a base pairing rule that exists with these A's, T's, G's, and C's. Okay, so this is called the complementary base pairing rule. The rule is really simple. All you have to remember is that A is always going to bond with T. The other part of that rule is that G is always going to bond with C. So if you're using a DNA molecule and it's double-stranded and you get a code like this, right, and that's one strand, then using this complementary base pairing rule that you see here, the A has to be paired with a T on the other side, C has to be paired with G, and so on. Okay, so it's not difficult. There is, though, this rule, and you have to apply that rule. All right, now let's look at how these nucleotides are strung together. Hopefully today you got to, or yesterday you got to the DNA model activity where you actually put these together. And I just noticed I left something off my diagram here. Hold on. There we go. So what you'll notice is that DNA is made up of 2 strands. Okay, so here's strand 1 and here's strand 2. And the strands are chains of nucleotides. Okay, so a chain is a strand. Okay, and that didn't turn out too well at the bottom, but just remember, a chain, when we say a chain, we're talking about a strand of the DNA molecule. A chain of nucleotides, and notice here's phosphate, sugar, base. Oops, there we go. Phosphate, sugar, base, phosphate, sugar, base, phosphate, sugar, base. Okay, so you've got these 2 chains of nucleotides. Here's the other side, right, phosphate sugar, and we're going to fill in these bases that go at the empty spaces here. Okay, and sorry about my diagram, I'm always bad on this other side of the molecule. Now, you'll notice before we fill in those bases that DNA has two repeating segments because there are repeating parts of the nucleotides. Okay, and there's one on this side, there's one on this side over here. This is called the backbone. Okay, backbone of the molecule. It's this repeating phosphate sugar segment that you're seeing. So here's a phosphate, there's a sugar, phosphate sugar, phosphate sugar, phosphate sugar. And DNA has another backbone on this side as well, right? This repeating phosphate sugar backbone. So those are the backbones, and because you have two chains, you have two backbones in DNA. All right, so let's go on now and look at what do we pair with these using the complementary base pairing rule. A, we're going to pair over here with T, right? Okay, so T goes with A. The next one we have to pair is T with an A here, right? And this is, we're just following along with your notes. G is going to go with C, right? And C is going to go with G, just like that. All right, so you've got, this is basically what the molecule of DNA looks like all laid out. Now the code on the DNA molecule is written in the order that we put these A's, T's, G's, and C's. So you'll notice, remember on the last slide, I just made a random strand of DNA, and the order of these bases in the DNA molecule is going to determine the code. Because every other part of the nucleotide stays the same, the only thing that changes is which bases we put in which order. But other than that, the last thing that you have to remember about the DNA molecule is that it's also often referred to as the double helix, right? Which is a twisted ladder. So if you kind of use your imagination, it looks like you've got the sides of a ladder here, that's the backbones, and then where these bases bond together, those are the rungs of the ladder, okay? And those rungs of the ladder are the bases here. Oh, one last thing. Forgot to talk about the bonds that hold these together. Okay, the bonds that are going to hold our A's, T's, and G's and C's. These bonds are hydrogen bonds. Hydrogen bonds are weak. And it's really important you bear that in mind, because if this molecule is going to fall apart at all, it's going to fall apart right at those hydrogen bonds. Okay? And we're going to use that property when we get to talking about replication. So that's DNA structure. Double helix, twisted ladder. So this molecule would get twisted. And you've got your chains of nucleotides.